Good morning. As you know, we are uh, continuing the chapter Principles of Inheritance and Variations. Okay. Now, in last class, I will cover. Sorry. Uh, I covered the topic human genetic disorder in which color blindness and sickle cell anemia. Now, today we will continue from another genetic disorder, phenylketonuria. Phenylketonuria is a metabolic inborn error. It is inherited from autosomal uh, inborn error in metabolism. Inborn error in metabolism. Okay. And it is due to autosomal recessive trait. Autosomal recessive trait. In phenylketonuria, what happened? The enzyme which converted uh, phenylalanine into tyrosine is lack. You know that some uh, enzymes play a great role in metabolism, overall biochemical reaction in our body, which is happening. So, one enzyme which is converted phenylalanine into tyrosine. In this phenylketonuria, this enzyme which converted phenylalanine into tyrosine is lack. So, what happened? This phenylalanine accumulated in body because it is not converted into tyrosine and changed into phenylpyruvic acid. Phenylpyruvic acid. And phenylpyruvic acid is very dangerous. It causes mental retardation. So this is the inborn disease, but inborn error in metabolism, but it is related with genetics, human genetics, and it is called as genetical disorder also. Next genetical disorder, we will take thalassemia. 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 Thalassemia is an autosome linked recessive blood disease. Autosomal Autosomal linked means this disease is related with autosome chromosomes. You know that in human genome two types of chromosomes, 44 chromosomes are autosome and two pairs, one pair is known as sex chromosomes. So this is related with autosome linked recessive disease, means when genes are in homozygous condition they are recessive. If one gene is healthy and one gene is of this disease, so that will not be inherited and that is not a disease. Sorry, inherited तो होगा लेकिन ये disease show नहीं करेगा, it will not show. In this disease both parents can be carrier if they contain one gene. So it in recessive form it is not show itself, but if both the parents, father and mother, both have this disease, so defective genes from one from mother and one from father, if it is inherited in the child, so that will be affected, it will causing the thalassemia. तो ये रिसेसिव डिजीज़ है, इसको होमोजोमस. नाउ ब्लड, हाँ, this one, it is transmitted from parents to uh, offspring. It is related with RBC. In this disease, hemoglobin is not synthesized. Hemoglobin is not synthesized properly. As you know that sickle cell anemia in that cases is same, but on that cases pattern is change, means one point mutation is there. One amino acid is replaced by another amino acid, but here whole globin chain has been changed. So they in this disease, RBCs are small, pale, short-lived. In sickle cell anemia, RBC converted into sickle form, and it is very common in Mediterranean sea countries which are situated at the border of Mediterranean Sea. In thalassemia, we know that one of the globin chain is absent, or they reduce the synthesis. For example, 2 alpha chain and 2 beta chain. So if one or two alpha chain is absent, तो बीटा चेन accumulation is more, यानी चारों के चारों बीटा चेन हो जाएगी. इफ बीटा चेन इज एब्सेंट तो चारों की चारों अल्फा चेन या नहीं होगी. तो दिस इज द डिफेक्ट. तो on the basis of the chain affected, it is classified that alpha thalassemia and beta thalassemia. Alpha thalassemia and beta thalassemia. In alpha thalassemia, production of alpha globin chain is affected. Alpha thalassemia is controlled by two closely linked: production of alpha globin chain is affected. Here production of beta globin chain is affected. Clear? It is related with two gene which is called as HBA1 and HBA2. Oh no. HBA1 and HBA2 on chromosome 16. Here in beta chain, if they are reduced, तो beta chain in excess, beta chain बढ़ जाएगी. तो due to excess of beta chain, they form unstable tetramers, which is called as hemoglobin H. Hemoglobin H. Clear? And which have abnormal oxygen dissociation. Here it is controlled by a single gene HBB on chromosome 11. So remember it, alpha thalassemia is related with two gene controlled by two gene present on chromosome 16, and beta thalassemia is controlled by single gene HBB on chromosome 11. Here when alpha globin is reduced, so excess of beta chain increase its number, and this is the and its form unstable tetramer which is known as hemoglobin H. Clear? In 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 beta thalassemia, formation of beta chain is stop or few beta chain forms. In both the cases, एक्सेस ऑफ अल्फा चेन इज़ देयर बट डू नॉट फॉर्म टेट्रामर्स. मतलब यहाँ जैसे भी अल्फा चेन नहीं थी, बीटा चेन बन गई तो यहाँ टेट्रामर बना जिसको HBH कहते हैं. लेकिन यहाँ अगर अल्फा चेन एक्सेस होती भी है तो दे नॉट फॉर्म टेट्रामर. इफ़ दे नॉट फॉर्म टेट्रामर तो दे बाइंड with RBC membrane and damage it. And high concentration they form toxic aggregates. They form toxic aggregate. तो ये होता है. So child will not live long because there is no cure. It is a genetical disorder. So these are the human genetical disorder which is related with genes. Now we will continue the disorders arising from chromosomal abnormalities. यहाँ पर क्या था, genes abnormalities थी कि gene change हो रहे हैं. Now we will move to chromosomal abnormalities, है ना. Chromosomal abnormalities. Okay, chromosomal abnormalities. These disorders are due to abnormal chromosomes number. It means abnormal chromosomes number. In normal chromosomes number क्या होते, 44 plus XX and 44 plus XY. Normal female and this is normal male. Okay. Now disorders are first autosomal abnormalities. As you know that two types of chromosomes, these are known as autosomes, and these are sex chromosomes. The first autosomal abnormalities. Autosomal abnormalities, Down syndrome. First is only one, that is Down syndrome. Down syndrome, it is in condition they have 47 chromosomes instead of 46. मतलब 44 plus 2 46 होता है. ठीक है, 46 में दो तरह के होते हैं, मेल और फीमेल. तो 46 in place of 46 they change into 47. It is called as trisomic. It is due to trisomic for trisomic for chromosomes 21. Trisomic for chromosomes 21. Thus there are 47 chromosomes instead of normal 46. For example, uh, 45 plus XX होगा और 45 plus XY. It forms 47. It is a different from normal. So it is this is also known as Down syndrome is also known as triplo 21. मीन्स ट्रिपल है यानी वन एक्स्ट्रा है कहाँ पर 21 क्रोमोसोम्स पे. सच individuals are developed non-disjunction of autosomes. It is due to gamete is fertilized by normal gamete of the opposite sex, or a zygote is produced which is trisomic. So affected children are mentally retarded, prominent forehead, flattened nasal bridges, and they are uh, projecting lower lips, large protruding tongue, and the fifth finger is short with only one transverse crease. And the Downs babies are also associated with congenital defects in heart and other organs. Mothers over 35 and those who have had one Downs babies are at the highest risk. Now, another abnormality is due to sex chromosomes. Abnormalities in sex chromosomes. Sex chromosomes and abnormalities इसमें first is Klinefelter's syndrome. Second, Turner's syndrome. Klinefelter syndrome is categorized by trisomy of. It is due to uh, trisomic for sex chromosomes. यानी 3 हो जाते हैं XXY. So such individuals are overall masculine due to Y, presence of Y. That individual overall masculine characters. They have some degree of breast development and tend to have longer than normal legs. Such individuals are often mentally defective and they are sterile males. Now second one is Turner syndrome. It is due to 46. Here is 47, 47, or here is 45. 44 plus x0, that is 45. Here is one more, here is one less. So another sex chromosomes is absent. So they are genetically, Turner syndrome is they are genetically sexless. They are immature female. They are categorized by poorly developed ovaries and underdeveloped breast. Another interesting sign is the presence of short fourth metacarpal and metatarsal bones. They are short statured. One female is in every 2500 birth maybe suffer from this syndrome. So these are the uh, disorders from chromosomal abnormalities in human being. Now we will start new topic of this chapter, that is the mutation. Mutation, as you know, as you study in junior classes, a sudden change in structure and number of chromosomes is known as mutation. So here is mutation where alteration of DNA sequences result in change in the genotype and phenotype of the organism, that is the mutation. So this is spontaneous or uh, may be induced. Spontaneous means अपने आप हो जाता है, या induce means ज़बरदस्ती कराया जाता है, artificially induced. तो type of mutation is which is in your syllabus as point mutation, frame shift mutation. Point mutation means is also a gene mutation. Gene mutation means at one fixed point. If something is changed, any change in nucleotide sequence nature can affect the expression of gene adversely. The mutant gene and the original gene are situated at the same fixed point. So it is called as point mutation. This type of mutation occurs in molecular level, usually at the time of DNA replication. Some errors are there, so that is the point mutation. We can give the example of sickle cell anemia, where the one nucleotide is changed, that change of the. Now frame shift mutation is caused by deletion or addition of nucleotide in a DNA segment. This genetic code is commaless. Both addition and deletion of nucleotide shift is reading frame of codes, so they are called as frame shift mutation. They are very harmful mutation. Gets completely changed. They change the sequence of amino acid in a protein. Uh, Human hereditary disease is caused. Muscular dystrophy is a frame shift mutation. So there are two types: deletion and insertion. Deletion, when one nucleotide is removed, uh, and here is added to the change. And next is one is frame shift, and next is substitution mutation. Substitution mutation. Okay. Substitution mutation, in these mutation, particular nucleotide of a DNA segment is substituted, replaced by another. So it is also two types: transition and transversion. Transition, those base pair replacement, where is purine is replaced by pyrimidines. Sorry, not it is transition. Purine is replaced by another purine. Transversion is purine is replaced by pyrimidines, or pyrimidine is replaced by purine. So that is the transversion. The sickle cell anemia is base substitution mutation because base in one of the gene involved in the production of hemoglobin. So, where is amino acid sequence for the normal and abnormal chain differ in the substitution of valine for glutamic acid, है ना. तो at one point तो in the abnormal polypeptide chain, so this minor change is caused the sickle cell anemia. Chromosomal mutation is not in your syllabus. Now genomic mutation, substitution, genomic mutation, uh, genomic mutation. Genomic mutation means whole genome is changed, मतलब whole set of chromosomes has been changed. Change in chromosomal numbers, the whole set. So there are two types, aneuploidy and euploidy. Aneuploidy and euploidy. Okay. Aneuploidy, aneuploidy means presence of chromosome number which is different of the multiple of basic chromosomes. Uh, this type of variation involves one or two chromosomes, not the entire set. That is the aneuploidy. It is again two types, hypoploidy, hyperploidy. Hypoploidy मोनोसोमिक एंड नलीसोमिक. इट मीन्स एन्यूप्लॉइडी मीन्स होल सेट में कुछ कम हो जाए. ठीक है, तो इट इज़ टू टाइप्स, हाइपोप्लॉइडी जहाँ कम हो जाए, हाइपरप्लॉइडी जहाँ ऐड हो जाए. हाइपोप्लॉइडी इज मोनोसोमिक when one chromosome is less than entire genome. Nullisomic when two chromosome is less than. Now hyperploidy. Hyperploidy is also two types, trisomic when one chromosome is added with set, and tetrasomic when two chromosome is added. Euploidy is variations, again now euploidy. It is the variation that involve entire change, entire set. Here is not entire set, it has a number of chromosomes which are add or minus. हाइपो में माइनस हो रहे हैं और हाइपर में ऐड हो रहे हैं. यूप्लॉइडी में पूरा पूरा सेट माइनस या प्लस होता है. फॉर एग्जांपल अगर एक सेट कम हो गया यूप्लॉइडी 2n minus n, so that is n, it is haploids. और जब ज़्यादा हो जाता है प्लस n मीन्स 3n, sometimes 4n, तो दे आर पॉलीप्लॉइड. डिप्लॉयड हैप्लॉइड तो हम जानते हैं, डिप्लॉयड इज़ नॉर्मल कंडीशन. जब व्हेन वन सेट इज़ प्रेजेंट तो डैट इज़ हैप्लॉइड. ठीक है, two set almost it is the normal condition, two sets should be present, but when these sets are increase in the number, that is polyploids. Polyploids are also two types, autopolyploids or allopolyploids. Autopolyploids means same set is repeated. जैसे सपोज ए है तो इसी का बढ़ता जा रहा है पूरा पूरा सेट. ठीक है, और एलोपोलिप्लॉइड्स जहाँ दो सेट हैं वो बढ़ रहा है. जैसे फॉर एग्जांपल F1 जनरेशन में ए बी है, अब ये पूरा ही ए बी बढ़ जाए, इट मीन्स ए एंड बी बी हो जाए, तो ये एलोपोलिप्लॉइड कहलाता है. तो now this is overall uh, chapter. One more information I want to give you, that the induced gene mutation. Gene mutation is due to some uh, agents, mutagens. मीन्स दे आर एजेंट्स फॉर म्यूटेशन, तो इसीलिए इनको म्यूटाजेंस कहते हैं. सम नेम यू हैव टू learn it, that is 5 bromo uracil, that is mutagen, nitrous acid, uh, uh, x-rays, ultraviolet rays, all radiations, they are defective, they are harmful for human being because they can change. So these are the mutagens. Now this chapter is over, and by tomorrow I will start a new chapter, that is the molecular, maybe molecular, or which chapter is that, molecular basis of inheritance.